My name is Laura Frost. I am an assistant professor at the University of South Alabama, and I am a plant systematist. I do phylogenomic research to study macroevolutionary patterns, and I'm specifically interested in patterns underlying diversification in biodiversity hotspots. My lab is currently working on pondweeds, a family of submersed freshwater aquatic plants that are known to readily hybridize, and we are interested in hybridization as well as the role of the North American coastal plain as a climate refugium in generating and maintaining North American pondweed diversity. My first experience with Arbor Biosciences was using one of the pre-made bait sets, Angiosperms 353, working with a group of tropical trees, Fresiera, that didn't really have any genomic resources available. So this universal bait set to capture 353 genes across any group of angiosperms was really essential in us being able to generate a phylogeny for the group. But as I've transitioned into this new study system, Potamogeton, or the pondweeds, one thing that we knew going into it was that previous studies had really struggled to resolve phylogenies due to low genetic variability among the loci that were available with universal primers and Sanger sequencing. We also know that there's hybridization as well as polyploidy. So we have some complex evolutionary history. That means that we really wanted to be able to capture the genes across different copies and from different parents, potentially within those hybrid samples. So a custom bait set really felt like our best option to be able to efficiently capture the loci and sequence at a depth that was needed to be able to phase reads for both polyploidy and hybridization. Interactions with the sales team were really great, positive. Everybody responded quickly. I very quickly felt like I had a whole team of people working with me and for me. So that has held up across kind of every level because I've since ordered more reagents for some of our high sensitivity samples. And yeah, in all of the interactions, everybody has been extremely helpful. And yeah, just really feel like you've got a team of people working for you. When we started this project, we knew that there were going to be some risky samples in there. We ended up generating data for 288 samples. 179 of them were herbarium specimens. One of them even predated the Civil War. So we had a lot of degraded DNA. We had older samples. So there were risks, low DNA concentrations, some very high sensitivity samples, and we did library prep and target enrichment in-house. So there were some risks we knew we were taking there with our samples, but even with those risks, out of the 288 samples that we sequenced, only four failed to assemble any data. And our probe set, we call it Pondweeds 350. We were targeting 350 genes, and for almost all of the samples, we were able to map reads to all 350 genes. The target efficiency was around 80% for the group, and for some of those older samples with lower concentrations, we still captured 340 plus genes of the 350. So knowing that we were taking some risks, that we had some lower quality DNA samples, we had incredible outcomes with our custom probe set.